The overall goal of the following experiment is to determine suitable conditions to study FtsZ polymerization and activity using simple assays and equipment. This is achieved by performing FtsZ polymer sedimentation assays under various conditions to find suitable conditions at which FtsZ polymerizes. As a second step, we perform light scattering assays to study FtsZ polymerization in real time. Next, we perform GTP hydrolysis assays to determine the polymerization-associated GTPase activity of FtsZ. Results are obtained that show which conditions are best suited for the study of FtsZ of a specific bacterial source based on sedimentation, light scattering, and GTP hydrolysis.
The techniques shown here are easy to perform in any laboratory and will provide a quick idea of the polymerization capabilities of specific FtsZs. We find that the choice of the right buffer is especially important when working with FtsZ in the presence of other proteins. Demonstrating the technique will be Eva Król, a graduate student from my laboratory.
To begin, add 5 microliters of 100 millimolar magnesium chloride and 12 micromolar of the bacterial proteins FtsZ and/or SebF into one of the chosen polymerization buffers that have various pH values and compositions, as described in the accompanying text protocol. Next, place the tubes into an incubator with a shaking function and incubate the samples for 2 minutes at 30 degrees Celsius and 300 rpm. Once the tubes have been thoroughly mixed, begin polymerization by adding 1 microliter of either GTP or GDP mixture from a 100 millimolar stock solution so that the final concentration